Oh, okay, this is recording now. Yeah, because clearly my husband doesn't know the words to Winnie the Pooh. So, this is the first of 2 screencasts looking at the process of sugar breakdown, so aerobic and anaerobic respiration. So to start with, our learning goal is we should be able to explain the chemical and energy changes that occur during the glycolysis phase of cellular respiration. The second 2 screencasts will look at a process called pyruvate oxidation and the Krebs cycle. And the success criteria, you will know you have met the learning goal if you can describe the general phases in glycolysis. I will not have you memorize every single step. I do, however, want you to know some of the important intermediates. And that's because they come back up in photosynthesis, and they're going to be something that if you're carrying on with biology, and God help you if you're taking grade 12 U Bio, you probably are, these are something that are going to come back over and over and over again. So we need to build a familiarity with them now. When I talk about important intermediates, I will stop and explain to you each step. And if you've downloaded the screencast viewing guide, you'll notice that the last page of that screencast viewing guide is a list of important intermediates and where they're found. So I'll tell you, include this intermediate, and I suggest you stop, pause the screencast, add it to that phase. Okay? So you should be able to describe the general phases and key intermediates. You should be able to explain the goal. And again, that's explicit in your screencast viewing guide, or the Cornell notes, whatever, I can't remember what I called them anymore. Describe the difference between anaerobic and aerobic processes, and I'll explain that in a second. And identify the differences in energy between starting and ending points in glycolysis and where energy is stored at the end of glycolysis. So the big thing we're looking at is, can you track the carbon and can you track the energy through these processes? And if you can do that, you've met the learning goal. So an overview of aerobic and anaerobic respiration. Key terms. You need to be able to tell me what aerobic respiration is. So aerobic respiration is, do you want to say hi, Sophie?
Hello.
Can you say hi?
Hi.
Hi. You just said hi to all mommy's kids. And technically the internet, as this goes on YouTube.
Yes. Okay.
Okay.
Let's go.
Go to bed. Mommy will be in a bit to give you a hug. Sorry about that brief interruption. So aerobic are pathways that require oxygen to occur, and anaerobic occurs without oxygen present. So the prefix "an" means none or no or without. So aerobic is oxygen requiring, and anaerobic is oxygen independent, or in some cases it can happen without oxygen. In other cases, it just flat out won't happen at all if oxygen's present. So this is a quick overview, and I will photocopy this little mini version, and then I have a placemat that we're going to look at when we get back to class that sort of goes through this. So glucose enters the cell and undergoes the process of glycolysis in the cytosol, which is another word for cytoplasm of the cell. So glycolysis occurs in the cytoplasm. Glycolysis makes ATP. The products of glycolysis move into the mitochondria, and there are 2 products of glycolysis that we're going to look at. There are electron carriers, which we'll talk a little bit more about in the next slideshow and in the next couple days. And then there's the actual sugar breakdown products. So both of those enter the mitochondria, where they are broken down into 6 molecules of carbon dioxide, and they're used to produce additional ATP molecules. And they also generate more of these electron carriers, which go into a process called the electron transport chain, which we'll cover together in class, and make ATP. So mentioned in the last screencast, there are 4 main steps in sugar breakdown: glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain. The 3 last ones are the aerobic steps. So they will only happen in the presence of oxygen. Glycolysis, which we're going to talk about today, is anaerobic. So glycolysis happens in all cells regardless. The 3 last steps, pyruvate oxidation, the Krebs cycle, and the electron transport chain, only happen in the presence of oxygen and only happen in eukaryotic cells that contain mitochondria. So a review of grade 11, if you can't remember, There are two different cell types, eukaryotic cells and prokaryotic cells. Eukaryotic cells have a nucleus and organelles, membrane-bound organelles. Prokaryotic cells do not. They have a nucleoid region, they have ring-shaped DNA, and they have no membrane-bound nuclei. So that would be things like bacteria and other archaea. So glycolysis in more detail. It was mentioned in the last slideshow, I think. Greek words, I'm getting confused, I think, between what I was doing in the viewing guide and what I've got here. So glycolysis comes from the Greek roots glykos, which is sweet or sugar, and splitting. It translates literally into sugar splitting. The main function of glycolysis is to convert glucose into two smaller molecules of pyruvate. So it takes a 6-carbon glucose and converts it into a 3-carbon molecule of pyruvate or pyruvic acid. And it does make some ATP. And in aerobic respiration, that pyruvate can be further broken down. So it generates the pyruvate that's needed for the more energy-rich step of aerobic respiration. It's a 10-step pathway, and it can be broken down into 3 sort of broad phases: preparation and activation, the actual act of splitting the sugar, and then an oxidation and energy yielding step. Keep in mind that we talked about redox reactions before. So things that are reduced gain electrons. They gain a hydrogen or lose an oxygen. Things that are oxidized gain oxygen, lose electrons, or lose a hydrogen. So oxidized molecules like CO2 have had much of their energy extracted, whereas glucose molecules are highly reduced. So there's lots of oxygens, there's lots of hydrogens in glucose. When you get to the carbon dioxide, it is the maximum number of oxygen atoms that can be stuck to a carbon. So it is completely oxidized. You couldn't add any more oxygen to it if you tried. You couldn't take any more energy out of it if you tried, really. And that's kind of evidenced by the fact that CO2 is really inert. So the main role of glycolysis in most cells is to generate that pyruvate, which goes on into the Krebs cycle. In simpler organisms, it will be the be-all and end-all, but they don't have the same kind of energetic demands. And so this is important because the amount of energy that's generated in glycolysis, the amount of ATP that's made, is much smaller than those aerobic pathways. So we'll take a quick look at this animation if it decides it wants to work. Maybe it won't. Where are we going here? Why don't you want to open? I want that slideshow to open. It thinks it's open. Oh, little Mac. I wanted this open. Okay, so let's try this.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, so what I'd like you to do is keep in mind the way they represented the glucose molecules in that overview. So they show glucose as basically a series of 6 spheres, and that's how we can think of them for the purposes of this demo. So when you're representing the molecules, drawing the series of six spheres is sufficient. So the net reaction is shown here. Okay, so the first 3 broad steps are activation and preparation. It's helpful to think of needing to split your glucose molecule exactly in half. And in order to split your glucose molecule exactly in half, you want to create a symmetrical molecule, and you need to have it look exactly the same. So ATP is actually input in the beginning stage of glycolysis. You actually need 2 molecules. And what that does is it adds a phosphate group to carbon 6 and carbon 1. And the glucose molecule is rearranged to form fructose 6-phosphate. And if you look at the structure of fructose 6-phosphate is a 5-carbon sugar. So it's got 2 sort of carbons sticking out on either side of a central oxygen. And what that means is that by the end of step 3, the glucose has been converted into a fructose 1,6 bisphosphate or biphosphate. So it ends up having a phosphate group here and a phosphate group here. So it's quite simple to kind of cleave that sugar in half, and you end up with 2 sort of similar molecules by the end of step 3. The enzymes involved in those reactions. Now, I don't expect you to know specific enzymes associated with specific reactions, but what I would like you to get to do is get some sense of where these names come from. So the prefix hex usually means 6, and a kinase is an enzyme that adds a phosphate. So a hexokinase is something that adds a phosphate to a 6-carbon sugar. Phosphoglucoisomerase is phospho is phosphate, gluco, and isomerase is, like we know an isomer is something with the same molecular formula and a different structural formula. So a phosphoglucoisomerase is something that rearranges the structure of a phosphoglucose. And then phosphofructokinase, again, the kinase is something that adds a phosphate. So in this case, it's going to add a phosphate to a phosphofructose. Okay. The next step is sugar splitting. So when you've got that sort of symmetrical fructose 1,6 bisphosphate, you can split the sugar in half. So the fructose 1,6 bisphosphate is split into 2 3-carbon compounds, dihydroxyacetone phosphate, which we're going to abbreviate DHAP, and glyceraldehyde 3-phosphate. So you should, on your list of compounds that you're tracking, have fructose 1,6 bisphosphate, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. So please make note of the compound, where it shows up in the cycle of glycolysis, but leave some space because you can actually add to that list where it shows up in photosynthesis, because you'll see some of these compounds will reappear then. And give a quick sketch, but use the sketch method that was done in the animation. So just show them as little dots. So the fructose 1,6 bisphosphate, let's see what I can do with a mouse here. You'd have your O, and it's just going to look something like this. So we've got a carbon here, a carbon here, a carbon here, carbon here, carbon here, a carbon here, and a phosphate group over here, and a phosphate group over here. The other way you could show this is just simply the line with the 6 dots. Apparently that last bond is longer. And your phosphate here. And your phosphate here. I'm actually pretty self-satisfied there. That was some impressive mouse work. Let's do blue. So basically what I'm doing in this lysis step is I'm cleaving my sugar here. All right. And you can see it better in the ring structure. The molecule isn't completely symmetrical because it is in the ring in the cell. So because it's shaped like a ring in the cell, these dihydroxyacetone phosphates and the glyceraldehyde 3-phosphate actually have slightly different structures. Okay, so the dihydroxyacetone is the ketone version. So your double bond O is here, whereas in glyceraldehyde 3-phosphate, your double bond is here. So we do have 2 totally different molecules. So dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are made. So glyceraldehyde 3-phosphate, your P is here. And dihydroxyacetone phosphate, your P is here, although it doesn't really matter. So you've got 2 different compounds, and I would put each of those in your note. But like I said, just draw them like this. You don't need to be able to draw the exact molecule. What you need to do is have a sense of how dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are different, and that they are different. So only glyceraldehyde 3-phosphate moves on. So we've got this kind of 5th reaction or 4B reaction, depending on how you want to think about it, where a dihydroxyacetone phosphate is converted by an enzyme into glyceraldehyde 3-phosphate so it can move on in the pathway. Hold on a second, my kid is crying. So, moving on. Steps 6 to 10 are the payoff phases of glycolysis. So far, by the end of step 5, we've invested 2 molecules of ATP. So we've put energy in, but we haven't gotten any energy out. So steps 6 to 10 are the phases where we start to see some energy yield. And you'll see that fairly consistently in the next set of reactions and in photosynthesis as well. You have to invest some energy to get something that's a good energy producing compound. So when we get into photosynthesis and we look at the fact that the plants take the time to build the sugar, it takes them less ATP to build the sugar than they actually get out breaking it down through this exact same process. So the second half of glycolysis is the payoff. We produce ATP and NADH. Remember that NADH is an energy carrying molecule or an electron carrying molecule, and right now that's all I want you to understand about it. So in reaction 6, a phosphate ion is added to G3P, producing 1,3-BPG or 1,3-bis-phosphoglycerate. So again, we've got our 3-carbon compound. And this is glyceraldehyde 3-phosphate. So right now, when we start, we've got our oxygen here, and we've got a phosphate group on this carbon. Okay. By the end of the 6th reaction, we're going to take that aldehyde and convert it into an alcohol that we then stick a phosphate group on. We've got, I'm not showing the alcohol that's on this middle carbon, but there is an alcohol there. And we've got a phosphate group over here on carbon 3. All right. So we start with this. By the end of step 5, we have the 2 molecules of glyceraldehyde 3-phosphate. So there's my aldehyde and my phosphate on carbon 3. And in reaction 6, we take that and we add kind of this random phosphate group that's just floating around in the cytoplasm onto the first carbon, creating a molecule of 1,3-bis phosphoglycerate. So again, the bis means 2, phospho is phosphate, and the glycerate is our 3-carbon compound. So 1,3-bis phosphoglycerate, voila. And when that occurs, because this carbon is being oxidized, we're adding a hydrogen, a molecule of NAD+, we're breaking a bond. It's oxidized, trust me, it's late. A molecule of NAD+ is reduced. So this carbon is oxidized, a molecule of NAD+ is reduced, and that produces this NADH molecule, which is an electron carrier. So we take the electrons off this carbon and stick them on a molecule of NAD+ to create NADH. The next reaction, reaction 7, one of the phosphates is transferred from 1,3-BPG onto an ADP. And what we call that is substrate level phosphorylation. And these terms are going to become important because we'll contrast substrate level phosphorylation with oxidative phosphorylation, which we're going to learn about on Tuesday, or if you're watching this in a successive semester, whatever day you aren't watching this. So we start with a 1,3, stop laughing, go deal with the currently crying baby. Okay, I think he's hungry. I love that I could conceivably not re-record these, and my husband, my son could be in grade 12 watching these screencasts while listening to himself cry at 6 months old. Okay, so by the end of reaction 6, we have this 1,3-bis phosphoglycerate. What's going to happen is we are going to pluck one of the phosphates off this and stick it on a molecule of ADP. And I'll go over in class in a little bit more detail what ADP is. But suffice it to say, it's an adenine nucleotide that has 2 phosphates stuck to it. So we're going to take this ADP, we're going to pull one of these phosphates out and stick it on that ADP. So now it becomes ATP, all right? ADP is adenosine diphosphate. ATP is adenosine triphosphate. So I'm just plucking that phosphate off and sticking it on this guy. And it's called substrate level phosphorylation because it happens in an enzyme. It is a substrate, okay? And that's going to be a contrast to oxidative phosphorylation, which you haven't learned about yet. In reactions 8 and 9, the 3-carbon molecule is rearranged to produce a molecule of phosphoenol pyruvate, short for PEP. So if you could please make a jot note about phosphoenol pyruvate in your book, that's important because we will revisit phosphoenol pyruvate in the Calvin cycle, so in photosynthesis. And then finally, reaction 10 removes a high-energy phosphate from phosphoenol pyruvate, creating an ATP, again, substrate-level phosphorylation. Now I'm enjoying writing. But clearly not worth the time it took me to write substrate. Okay, so creates another molecule of ATP via substrate level phosphorylation. Now keep in mind, all of these energy yielding steps are actually happening 2 times per glucose. We will abbreviate glucose as gluc. Okay, so each of these steps happen twice per glucose because you've broken 1 glucose into 2 of these glyceraldehyde 3-phosphate molecules, which then become 1,3-bis phosphoglycerate, and so on. And that is the end.